Well, everyone, welcome back together. See, we still have 48 people, which is remarkable. I've been going from one to the other session. I saw a lot of, first of all, engagement, the complementarity of people's strengths is just, stupendous. I saw a lot of commonality in the discussions, they obviously intersected across different areas from the acute neuroinflammatory effects to chronic ones. I also observed a lot of discussion of limitations, as you are, we consider what we can do, each point that we've come up with, I didn't hear any free sailing, oh, this is clear, this is easy, which is, one of the considerations as we go forward. I think there are, whenever there's a hurdle, there's an opportunity. And we have to identify what those things, maybe the low-hanging fruit, but also, what some of the mid-hanging fruit is too, as we go forward. And I think the other observation I just shared in the room that I was just in, we're in a very reactive, an acute reactive phase right now. All of us are, all of us have seen our lives turned upside down, and also want to help. But we also have to consider the risk reward of pivoting our programs towards a new target, and with the possibility that that target disappears, and then you're left holding the bag. That's what happened with SARS the first time round, but this is clearly a different illness, and I think we'll all have to just consider that as we reorient our programs in this direction. Maybe we have a lot of people here. I think you can raise your hand. Let's open it up to other summative comments before we talk about maybe next directions.
I don't know. There isn't a raise your hand thing there. So
you go into participants. I'm sorry. If you open up participants, you can raise your hand on the participants.
So
and then,
all right.
And then I guess I see the person, so I'll call them out. Ofer, I just see first. Sadan.
Yes, hey, I'm Ofer from the neurocritical care. We were talking, the one thing that we were talking in the acute neurological injury section that is maybe very relevant to the bigger group is the fact that we're seeing these patients and sometimes we tap them, we have CSF, we have blood samples that I think would be relevant for many different types of research, but there is no single lab that is currently takes care of that CSF. So we know of CSF going into at least 2 different labs. And if anything, I think that this group could come up with a single lab that is willing to take ownership of those samples for a bigger sample and studying it.
Great, thanks. And by the way, this main room is being recorded, so we will have those comments recorded. Annabelle?
Okay, I was in the cytokines group, neuroinflammation group. I'll just maybe repeat some of the big questions we talked about. They're open questions, so we don't have answers. Does the virus get into the brain or not? Meaning, are we looking at first order or second order effects, or both? What are the primal viral reservoirs? What are the short-term and chronic neurological effects? How can we get samples and data from patients? This was a big point, that neuro is a lower priority than, say, things like respiratory failure. And so there are major hurdles to just getting the samples that we would want or need to make progress on some of these questions. Are there neurological subtypes? We've heard about various different symptoms, but are there sort of categories that we should be thinking about?
Great, thank you. And I was kind of remiss in not saying we have one person from each room give exactly that type of readout from your room. That would be wonderful. Andrew, your hand is up. Maybe you can do that for the psychiatric and neurologic room.
Well, I wasn't in that one, but
that's right. You were in the animal models. Yeah,
it does sound like that there is some consistency about some of the questions and some of the infrastructure. Our group felt that using either animal models or ideally human-induced pluripotent stem cells might be an excellent first step towards understanding which cell types are most vulnerable to infection and what in fact are some of the consequences of those of infection. Unfortunately, it brings us to the issue of infrastructure and getting access to BSL-3 facilities where some of this work can be done. It's been done with other viruses before, and these are some of the technological advances that allow us to answer some very basic questions that will then, I think, help inform some of the clinical issues. But the infrastructure for BSL-3, and then now I hear and I wonder the same thing, is are there a cohort of patients that are being gathered in our tissue samples or blood and CSF samples being banked, and is there some kind of a consistent clinical infrastructure that can then be accessed either to address some of the acute issues as well as the chronic, which I think will probably be where much of the action is what happens to people long term if in fact the virus is, we find, invading certain cell types in the brain and having long-term consequences. So that is a summary of what our group was interested in from the animal model side and the infrastructure and the iPSC cellular models side. And then I think that bleeds into these larger infrastructure issues as to what is going to be available to us to conduct the kinds of experiments that would be ideal in this situation.
So I'm going to jump in here because this is Kathy Grunling, because we're essentially out of time. I do want to tell those of you at Emory that Mike Zwick has organized 4 different working groups around collecting samples and patient data from these patients. So if you're interested in being involved in that, you can email him to be part of that working group. One thing we do need to decide before we end is the best way to move forward. And I'll tell you that previous Blue Sky groups have resulted in a shared listserv where you can exchange ideas. You can decide that you want to come back together again in a few weeks, especially each breakout group. We can do that independently of the larger group. But you need to let us know what you want to do. Bob, do you want to add anything to that?
No, just to say that go ahead and send us an email, copy me, Kathy, and
Lauren.
Mark Daniel.
And Lauren James, who's the organizer.
And Lauren, yeah. And we will collate all your responses. We didn't have a chance to get a lot of readout from, I know, even the biggest room, the psychiatric and neurologic room and the acute room. So send along your impressions and we'll collate them, and also your suggestions as to what you would like to do going forward. But I think we have a pretty good idea. I think it was just a great idea, Kathy, of having staggered breakouts so that we can sort of cross-pollinate and not be conflicted in which room we want to go into. And then think towards, getting a deliverable. We have to have a deliverable from this, or it's been just an exercise.
Well, we'd hope the deliverables would be joint papers, joint projects, joint grant submissions. That's kind of the goal of these groups.
Yep.
But of course, anything tangible like that is welcome.
All right.
Thanks for all your participation in this group, and do email any one of us and we'll put it all together and try to come up with some next steps. Thank you,
everybody.
Thank you.
Thank you.
Thank you.
Thanks.
Thank you.